disgusting discoveries to share with you guys today. So we're going to go back in time. This is going to be a paleontology talk, if you hadn't already guessed that from the title. And we're going to go back in time to the late Jurassic period, about 150, 152 million years ago, when the Morrison Formation was being deposited. Now, today, if you were to go out on Mack Ridge, out in Kokopelli trails, you would see vast expanses of the Morrison Formation. It looks like this. You have these beautiful painted red and gray marl shales and mudstones, as well as hard, resistant sandstones that are great for rock scrambling and just getting out with the kids and kind of finding things in the landscape. Now, this is the Jurassic Park of North America. The Morrison Formation is home to some of the most iconic dinosaurs from North America, from our state fossil of Stegosaurus to the giant plant eater Apatosaurus and the ferocious theropod Allosaurus. And we find all of these dinosaurs in the Morrison Formation in Western Colorado and in McInnis Canyons National Conservation Area. Now, in the Morrison ecosystem, we had rivers that were draining from the highlands that were located back in the Jurassic period in the southwestern United States. They were draining to the east and upwards to the north. They were emptying into this shallow inland sea that stretched through parts of Canada and parts of what would eventually become Montana. And in western Colorado in McInnis Canyons, we see evidence of these streams. We see evidence of them in the sandstones and in those painted mudstones. Now here's McInnis Canyons National Conservation Area, and our story, even though the Morrison Formation is found throughout McInnis Canyons, our story is going to take place here at the Mygammore Quarry over in Rabbit Valley. Now the Mygammore Quarry has been a big part of the museum and the museum's both research and outreach program for decades. We have spent over 30 years excavating fossils from this site, and what is so amazing about the Mygammore Quarry is we're still learning new things from it, which is wonderful. We have a radiometric date for the age of the quarry. It's about 152 million years old. And you can see that the quarry is in these gray shales and thicker mudstones, and it's quite a large area. You can see where we have, in this picture, all of our tents kind of clustered to one side, but it goes all the way over to this ridge here behind the equipment trailer. And this is a dinosaur bone bed deposit. What do I mean by bone bed? A bone bed is a very dense accumulation of bones. And in this case, they're in these shales and mudstones. Now the shales and mudstones are kind of a light to medium gray. And here on the left-hand side of the screen, you can see a bone starting to be excavated from the quarry. And the bones tend to be darker gray in color. Now, excavations of the quarry began in 1984. The quarry was actually found by a group of friends who were just out hiking in Rabbit Valley enjoying the National Conservation Area. They came across some bones coming out of the ground, and they did what everybody should do when they find bones. They called the museum, they called the Bureau of Land Management, and we went out and took a look. And curator of paleontology at the time, Lance Erickson, was very impressed with what he saw in 1981, but it wasn't until 1984 that the museum was able to come back and start excavations. And we've been excavating at the quarry almost every summer since then. We've recovered thousands of bones and teeth from the quarry, as well as lots and lots of plant material. The plant material isn't hard like the trees at Petrified National Forest. Instead, they're carbonized remains, so they're kind of like charcoal in the shale. And so you can sometimes see really cool things like pine cones or fronds, but a lot of the times it's just this kind of broken up bits of plant hash. Now the quarry is interpreted to have been near a waterway, but not in a waterway. So there would have been a river nearby. This would have been the floodplain that was near the river. The Morrison would have had a very wet, dry climate with periods of long droughts, as well as lots of wildfires, kind of like we see in the Western United States today. And it would have had standing water in this muddy area near the river during the wet season. And the three most common dinosaurs we find at the Mygammore Quarry are big herbivores like the sauropod Apatosaurus, here you can see at the top of the screen. Lots and lots of the Theropod, the meat-eating dinosaur Allosaurus, as well as the little ankylosaur, this little armored dinosaur called Mygammore that was first discovered at the Mygammore Quarry. Now, back in the Morrison's depositional time, back in the Jurassic, the stream would not have looked like the Colorado River does today, and it would not have looked like, say, the Platte River that runs through Nebraska. It would have looked more like a lot of the rivers we see at high latitudes today, like these anastomosing streams. This one, I think, is from Sweden, where you have multiple river channels and lots of floodplains. But between the river channel, instead of having sandbars like we see on the Platte River, you would have had more floodplain in between the channels. And the river would hop back and forth between these channels depending on flow and speed of the flow. And it was a really great area to grow lots of plants, and it was a great area to be a dinosaur and to make your living, to either eat plants or eat other dinosaurs. Now, if you were to hop in your time machine, you would see something that looks like this, sorry. You would see a river system, you would see lots of dense vegetation near the river, and then the vegetation gets a little sparser as you go out from the river. The forests of trees would have been made up of conifers and cycads and tree ferns. There would not have been any maple trees, there would not have been any sycamore trees back then. Flowering plants really weren't very common on the landscape, and even things like grass would not have been very common on the landscape. Instead, the ground cover would have been things like mosses and ferns. Now, in the wet season at Mygammore, you would have had occasional standing water like this, and scenes like this might not have been all that uncommon. You would have had large plant eaters coming down to drink or coming down to eat, and of course, following these herds would have been predatory animals, would have been predatory dinosaurs like these Allosaurus here, who are very excited that they're about to get a meal. The Apatosaur is less excited. But in the dry season, the scene might have looked a little bit more like this. The water would have evaporated away, would have dried out. The mud of the soil would have dried out into the sun-baked earth that you can see on the screen. And you would have had rotting dinosaur carcasses hanging out from animals that had either died sometime earlier in the year or had come down to drink, not found water, and died of thirst. And you would, of course, found predatory dinosaurs fighting over whatever scraps of food were left in the ecosystem. So during times of drought or times of large ecological stress, the Morrison Formation is famous for some of its evidence of wildfires and these severe droughts. And we find evidence for these things, for these stressed ecosystems, as well as better off ecosystems during the wet season at Mygammore. Now, both of these pictures of these two dinosaur paleo art panels that I've just shown you were both commissioned by the museum to represent Mygammore specifically. And so now I'm going to tell you some of the research that's gone into creating these dramatic artwork panels. And this talk is not really about a new dinosaur that was discovered or you know, new, large, amazing, giant fossil femurs. Instead, this talk is kind of going to be about how dinosaur carcasses decayed and the transition between being a dead animal on the landscape and becoming a fossil that paleontologists would come find later. So the traditional story of how dinosaurs become fossils looks something like this, right? You have a dinosaur that dies, it falls into a waterway like a river, it gets quickly buried with sediment. The sediment grows on top as more and more floods occur and deposit more sediment. Those rock layers compact down, groundwater comes into the system and fossilizes the bones. Then the bones later become exposed by erosion and are found by a paleontologist. This is the traditional scenario that is often used to describe how dinosaur fossils are formed, but this might not be what was happening at the Mygammore Quarry. And the more that we're researching what was going on at the Mygammore Quarry, the more detailed window we're getting into just how dinosaurs were becoming fossils. So the dinosaurs at Mygammore were their bodies were left on the surface for a long time. And the longer the bodies are left on the surface before they're buried, the greater the risk of them being damaged, either something steps on them or the sun bleaches out the bones, or the greater the risk that something is going to mark up the bones. You have a scavenging predator that comes in and disarticulates a bone that still has a little bit of meat on it, and its teeth kind of glance off the bone and make some marks on there. And what we're learning from Mygammore is how prevalent some of these marks were. Now, marked up bones from Mygammore have been known for a long time. This large bone in the center of your screen, this is one of our touchable exhibits at the Dinosaur Journey Museum. This is a pelvic bone from a large Apatosaurus. And you can see that on this end, there's some things on this bone that shouldn't be there. There are these deep grooves with lines inside those grooves. We also find bones with deep, smooth-walled holes going through them where holes should not be. We find these furrows that have scrapes all along inside the furrows. We find these little donut marks. We even find these very shallow parallel lines. We find lots and lots of bones from the Mygammore Quarry that have lots of different types of marks on them. So what our research crew wanted to do was to look at every single bone, every single side of every single bone, and survey every single mark we could find at Mygammore Quarry to get an idea of what was going on the surface of the ground at Mygammore to these bones before they entered into the fossil record. Now, lots of things can make marks on bones. You can have large herbivores coming in to drink at the waterways, and they trample whatever carcass is there and smash things up, break bones, push them down into maybe some of the loose soil or the muck if it's been wet. And then you have, predators that are gonna not pass up a free meal if they come by one. You can have the sun and other elements bleaching out and weathering the bones at the surface, as well as lots of little critters like bugs and other invertebrates that will feed on rotting flesh, right? Carrion insects for one. Now, what we did in order to find these bones is we assembled our crack team of researchers. So here you have myself, my colleague, Dr. Stephanie Drumheller, and then our two CMU interns, Anya and Miriam. And what we did was we physically looked at every single bone that we could get access to at the museum from Mygammore Quarry. So we looked at 2,368 bones. We excluded any bones that weren't on the premises, they were out on loan. We excluded bones that were still being prepped in the lab because their surfaces weren't clean yet. We did not look at the teeth, we bypassed the teeth. And then we also didn't look at everything that was on exhibit. We looked at a few things that were on exhibit, like that big dinosaur pelvic bone I just showed you, but most of the stuff that's on exhibit, we didn't include in the study. But still, we looked at a few bones. We had a pretty good data set to go with, so we decided to move forward with our almost 2,400 bones that we looked at. This took us about 2 years to complete. Actually, it took about 1.5 years to complete the actual physical part of looking at all the bones. And in order to find these very shallow, very subtle marks on the bone, you had to do what you're seeing us doing in these photos. You had to shine either a flashlight or look under a microscope at a very low angle and physically look very closely at each bone surface. Now, we wanted to know was, one, what types of marks were there. We knew that what some of the marks that had previously been identified were, but we wanted to know all the different types of marks that were there in the fossil assemblage. We wanted to know what those marks could tell us about what was going on in the ecosystem in the late Jurassic period. And we wanted to know how fieldwork at Mygammore might be messing with the preservation of these small subtle marks. Like a marked up bone is not always the prettiest bone. And sometimes those ugly bones don't get collected. So what we wanted to do is we wanted to look at our fieldwork protocol and see if we were getting an accurate estimation of the marks in the assemblage. So let's see who was there first. So of the 2,300 some odd bones that we looked at, 884 had at least one mark on them. Many bones had lots of marks on them, and many bones had multiple types of marks on them. This was about 37% of our whole data set had at least one mark on the bone. And What were those marks? Well, we found 2,161 individual marks, and the largest piece of that pie were marks from the teeth of theropod dinosaurs. The second biggest chunk of the pie were marks from invertebrate animals, so animals like snails and insects. And then the smallest piece of the pie was everything else. Everything else from abrasion and trampling and breakage on the bones, that was everything else. The two most common types of marks that we were finding on every single bone from every single type of animal were invertebrate traces and theropod bite marks. Now, of the 2368 bones that we looked at, 685 had at least one theropod bite mark. 383 had at least one invertebrate trace. That's almost 29% of our data set with a tooth mark from a carnivorous dinosaur, and 16% of our data set with marks from an invertebrate animal. These are some of the bite marks that we were able to identify from theropod dinosaurs. And as you can see, there are a lot of different types. Everything from a little pit where the tooth hit the bone but didn't go all the way through, to punctures where it punctured all the way through into the bone. You have scores, which are kind of like cut marks almost by the tooth scoring into the bone. And sometimes those scores have striations in them. Dinosaur teeth, theropod teeth, have serrations on them on both sides, like a double-sided steak knife. And when you pull a steak knife through, say, a stick of butter, you get a big groove with lots of little lines inside of it. Turns out you get the same mark when you pull a theropod tooth through a dinosaur bone. So we get a lot of these striated scores. Sometimes we get big grooves that are wide and well excavated by the teeth. And then sometimes we get these fluted striation marks. Sometimes when that theropod tooth hits a bone, it glances off the side. It doesn't quite go straight down point first into the bone. And when it glances off the side, those individual serrations will leave individual grooves on the surface of the bone. And those are called striation marks. Now, those striation marks turned out to be quite interesting. And once we started identifying those in the data set, Dr. Drumheller pulled me aside and said, hey, we should probably talk to a colleague of mine, Dr. Dominic D'Amore, because he has spent a large part of his career looking at the bites of theropod dinosaurs and Komodo dragons, both of which have those striated marks made by their serrated teeth. So both Komodo dragons and theropod dinosaurs have serrations on their teeth, and they make those striated marks. And what Dr. D'Amore was able to do in his research was come up with a mathematical formula to estimate a minimum body size for an animal that makes that kind of a mark. So we gave him a call and we told him what we were doing, and he was very excited and agreed to help us out with our research. So he took our data and plugged it in to his mathematical calculations, and we're able to group the different sizes, different minimum body lengths of the theropods doing the biting into 3 categories. So category number 1, we have a lot of striation marks that were made by an animal that was about the size of an adult Allosaurus, which is pretty handy because we get a lot of fossils of adult Allosaurus from Mygammore Quarry. Our largest adult Allosaurs out of Mygammore Quarry are about 30 feet long. So these are big Allosaurs. But it's normal for the animal to be about that size as an adult. Our second group of body sizes for these theropods that were biting all these bones is a smaller animal. The smaller animal would have been about the size of an adult Ceratosaurus, and we do get Ceratosaurus teeth from the Mygammore Quarry, or the size of a young Allosaurus, and we do get juvenile Allosauruses from the Mygammore Quarry. So both category number 1 and category number 2, we have body fossils from the quarry to support those animals of that size making these marks on bones. But wait, there's 3 sizes. So what's size number 3? Well, this is our big guy. Size number 3 is a large theropod. This is an animal that would have been much larger than any theropod we've ever recovered in over 30 years of digging from the Mygammore Quarry. We do not have any body fossils that would have ever given us a clue that there was an animal this size roaming about Mygammore 152 million years ago. But thanks to this study and thanks to the work of Dr. Dominic Mygammore, we were able to show that we have evidence that this big guy was roaming around the quarry, maybe taking a bite and moving on and happening to not die there. An animal this size would be about the equivalent of Saurophagonex from Oklahoma. Saurophagonex is only known from Oklahoma, not from Colorado. So we would never have guessed that an animal that size would have been in our quarry. But now we have evidence that there was a large animal doing some snacking at the Mygammore Quarry. Now, tooth marks on dinosaur bones are not new. We've known about them for a long time from the Mygammore Quarry and from other dinosaur fossil sites in the Mesozoic. The Cleveland Lloyd Quarry, the famous Wall of Bones at Dinosaur National Monument, Carnegie Quarry, and even up at Coma Bluff at areas like the Freeze-Out Hills. But previous research into dinosaur tooth marks on bones suggested that dinosaurs really weren't working the bone as a primary food source. They were more of making accidental marks as they were trying to feed on the good, soft, meaty tissue. Unlike what a mammal does, I don't know how many of you out there have dogs, but mammals tend to like bone a lot. A hound dog is never as happy as when it has a piece of rawhide on your living room floor. So in a mammal assemblage, we tend to see higher concentrations of bite marks on bones. So something along the lines of 13 to 37% versus a dinosaur assemblage where the normal was thought to be between 0 and 4%. Well, Mygammore is blowing this out of the water. At Mygammore, we have almost 29% of our fossils with a dinosaur tooth mark on them. This is a very large number. We weren't expecting it to be this high at all, but it is showing that at Mygammore, things might have been bad enough during times of drought that there was nothing left, and the dinosaurs were taking part of whatever carcass was on the surface because they were that desperate for food. Now, these theropod tooth marks are found throughout the fossils at Mygammore. They're found on sauropod dinosaurs. They're found on Mygammore pelta. And they're even found on different areas of the body. They're found on ribs and limbs where you have lots of good meaty muscle attachment. But they're also found on things that aren't necessarily as meaty, things like vertebrae. And I don't mean the parts where the ribs attach where there is some meat. I mean the round center, the centrum, the body of the vertebra. So places that don't have a lot of meat. We also find them on toe bones, not something that's necessarily known for having lots of meat. And we even find them on animals that aren't usually the first thing you would go after for a meal, other theropods. So we find these tooth marks on both the parts of the skeleton that are high economy, they have lots and lots of meat on them, and then we find them on parts of the body that have very low economy, they have very low relative nutrients, they have not a lot of meat attached to them. So here we have a toe claw from an Allosaurus and the centrum of another Allosaurus, and you can see both of these have theropod tooth marks on them, which indicates that they were getting to the carcasses late during the feeding, and probably that means that they were scavenging. They weren't, taking down the animal and eating it right away. And it also means that Allosaurus was probably feeding on its own kind.
Sometimes these predatory dinosaurs were even resorting to eating other giant predators, right down to the toes.
We commissioned Brian Ing to make this art for us as part of this study, and he was wonderful enough to include an animation, which is just awesome, that shows how desperate and how stressed out this Jurassic ecosystem was from time to time at the Mygammore Quarry. And this toe crunch is based on that theropod toe claw I just showed you. But theropods were not the only thing doing the scavenging on carcasses at Mygammore Quarry. They had a little help from the invertebrate animals. So let's look at what the bugs were doing. Now, invertebrate traces are very small. You have to really get your eye tuned when you're looking for them, but we found them throughout the assemblage on every type of animal and every type of bone. And we found 6 different kinds of damage from these invertebrate animals. Everything from scrape marks on the surfaces of the bone, these little weird donuts, big furrows all the way into the bone surface with scrape marks in them, these big chambers, small little boreholes, and even some weird, oddly straight-lined scrapes in the bone surface. Now, some of these were pretty easy to identify. So we have these furrows into the bone surface that sometimes have these scrape marks in them. We see these little donuts, and then we see these wandering trails of arc-shaped scrapes in the bone. All of these we have been able to observe in modern environments, particularly in modern laboratories, from these guys, from dermestid and clarid beetles. These are often used in museums, in taxidermy, as well as in archaeology to clean skeletons. And sometimes people forget and they leave bones in the beetle room a little too long, and the beetles will start to excavate into the bone. And that's how we know what types of marks these animals make. And these are the same types of marks that we're finding at the Mygammore Quarry. And so we can infer that that dermestid or clarid beetles were the ones doing the marks at this Jurassic fossil site. We don't have any body fossils of these beetles being present at Mygammore, but now we know that they were there, and they were quite prolific. We have some large sauropod bones that are densely covered in these pits and furrows. So they were there and they were abundant. Now, these groupings of small subcircular boreholes, this is a bone fragment from a limb bone of a sauropod dinosaur. We find clusters of these little boreholes together. I think this one bone had about 11 of them. And these are the right size and shape to be made by carnivorous gastropods. And we do have fossil carnivorous gastropods at the Mygammore Quarry, including Ampulovata. Ampulovata is found at the site. It's the right size. Its mouth parts are the right shape to make these small, shallow boreholes. And so we're able to assign this type of insect trait, or this type of invertebrate trace, sorry, to carnivorous gastropods. And in modern environments, carnivorous gastropods are known to bore into bone when it's available to them. Now, these large, deep ovoid chambers, notice that these are 1 centimeter scale bars. These large chambers are an order of magnitude bigger than those small boreholes I just showed you. So these things are oval in shape. They have kind of an irregular margin, and they go deep into the bone. They go at least 1 centimeter deep into the bone. What made them? We don't know. This is unknown invertebrate actor number one. There's another one. No known Jurassic invertebrate is associated with making a chamber this size and shape in bone. Now, in some modern archaeological settings, solitary burrowing wasps and wild bees have been known to make holes in bone this size and shape. But their groups, their lineages, don't evolve until the Cretaceous period. This is the Jurassic, so they weren't around yet. So we don't know who was making this hole, these types of holes. It was definitely somebody who was doing the same behavior that we see in these burrowing wasps and wild bees, but it was a different animal, and we don't know which animal. And we don't have any body fossils from Mygammore to help us out here. So we need to sample the rock record a little more and figure out who our mystery actor was drilling holes this size into the bones of dinosaurs. And then there's this scrape mark. When I first saw this scrape mark, it sort of looks like the teeth of a zipper, you know, before you zipper it up. I thought, this is really weird. It can't possibly be, you know, anything other than somebody hit a tool the wrong way. But then we started finding it on multiple different bones. And none of our tools would make a mark this size and this shape. So we started looking at invertebrate faunas that would have been the right size to make this shape, and we're up to unknown actor number 2. No known invertebrate is associated with leaving a trace like this. These are very, very weird. Nothing alive today makes a trace like this. No known invertebrates from the Jurassic period appear to make something this size and shape. These lines are very linear on all these different bones. They're always paired. We're unsure what this is. It's probably an insect, but we need to go back and find some more fossils and see if we can't identify who's making this mark. This is another mystery bug from the Jurassic period. Now, because at least the domestic beetles we can observe today in laboratory settings, we know how their living members, how long it takes them to lay an egg, how long the egg takes to hatch, how long those larvae pupate, how long it takes them to grow up in order to have the right mouth parts to make marks on bones. We can time this, and it takes about 5 months in living species. Now, their Jurassic correlates, we're not sure if they follow the same time frame on their development, but we can throw out a number here that for bones that have at least one of those marks, it would be about 5 months on the surface. Now, I mentioned that some of these bones are covered in those marks, and that's going to take longer at the surface. So this is a minimum estimate based on the domestic beetle colonies, but at least 5 months before dinosaur carcasses at Mygammore were getting buried. But we can also look at weathering patterns. Back in 1978, Kay Berensmeyer spent decades combing the wilds of Amboseli National Park in Africa, documenting bones laying out on the surface and how they were weathering away based on sun and damage from the elements. And she came up with a weathering stage system that categorizes how broken up and deteriorated bones are. Whereas weathering stage 0 would be the animal just died and the bone's still greasy, to, you know, weathering stage 4, like you can see at the bottom of the screen here, where the bone is cracked, it's splitting apart, parts of the periosteum have flaked away, and the bone is very brittle. I'm actually very impressed that bone D even made it into the fossil record. This thing was badly beaten up by the elements, and I'm pretty happy to have some evidence of it in our collections. Most of the bones that we've collected from the Mygammore Quarry are weathering stage number 1, which is at the top of the screen, and weathering stage number 2. But we do get a lot of weathering stage 3, and then the occasional weathering stage number 4. Now, based on Kay Berensmeyer's work, you know, she would have assigned certain time frames for each weathering stage based on the climate at Amboseli National Park and based on her own observations. We are pulling this back in time to the Jurassic period, which has a very different climate. It has a very different set of parameters. So take these numbers with a grain of salt and a wide set of error bars. But based on her calculations, this is indicating that carcasses are on the surface on average for weathering stages 1 and 2, 6 months to 6 years before they were being buried. Weathering stages 3 and 4 would have been 10, 15 years, much longer. We don't know whether conditions in the Jurassic would have sped this up or slowed it down. So this is just a benchmark number, but regardless, these carcasses were on the surface for a long time before they were being buried. They weren't being quickly covered by a flood of sediment. They were on the surface for a long time. And finally, because weathering stage number 4 is an ugly bone, how has our collection, how has our fieldwork collection affected what types of data we have in the Mygammore collection? Well, before 2016, small unidentifiable bone fragments like the ones you can see on the screen here were systematically not collected from the Mygammore Quarry. They were reburied at the site. And that's part of the reason for that is the museum's agreement with the Bureau of Land Management, who manages the Mygammore Quarry, is that when we're done, we need to revegetate the landscape. Well, fossils are rich in phosphorus, which can help plants take root and reseed the area. So there was a reason why these fragments were systematically not collected. They weren't able to identify them. They didn't appear to have any scientific value, but they did have value for restoration of the site later on. But once we started thinking about doing this study and we knew we wanted to look at ugly bones, we decided to change that. So in 2016, we instigated the nugget bucket protocol. So the nugget buckets are exactly what you see on the screen here. They are plastic buckets. They're 5-gallon painter's buckets. And that's where we've been putting all of these unidentifiable fragments from 2016 into today. And we have them kind of grouped in areas of the quarry. So the eastern part of the quarry, the northern central part of the quarry, and a large pit that we were recently working. And we sorted them into these buckets to look at and see, are they preserving any sort of bone surface marks that can impact our study? And between the field seasons of 2016 and 2019, we found an additional 59 bones with marks on them in these nugget buckets, which tells us that, yeah, we were missing some data by only collecting the nicer, larger, prettier stuff. So we knew we had to make a change, and we instituted new collection protocols for the Mygammore Quarry so that we could ensure that all the scientifically relevant data was being collected at the site. The new protocol states that bones have to have surface bone, cortical bone present on the specimen. So if it's weathering, if it's a small fragment that's weathering stage 3 where all of the surface bone is flaked away, we won't be able to find any surface marks. So those are okay to rebury. The bone also has to be larger than a postage stamp. The smaller you get, the harder it is to identify a bone surface mark, like a tooth mark, from say just a pattern of break at the edge of the bone. And then we also, with this study, created identification guides for our volunteers in the lab and for our field workers and families that join us at excavations in the field. So we created identification guides that have both photographs and 3D printed models of the different types of bone modification that we're finding at Mygammore to help train people's eyes to look for them as they're excavating. So I'm coming to the end of my time here. So let's kind of draw some conclusions from this gross study of dinosaurs decaying on the surface, oozing gross grease with bugs crawling in the carcasses, and theropods hungrily snatching any flesh that's left, even if it's their own kind. Well, conclusion number 1, carcasses were not buried quickly at Mygammore. They were staying on the surface for months to years, creating opportunities for large infestations of invertebrates to work the carcasses, to process the meat, to mark on the bones, and created opportunities for theropods to scavenge a free meal. And sometimes that free meal was scavenged by an animal that just dropped in and walked away, and sometimes those animals stayed behind a little too long and became part of the assemblage themselves. We also were able to identify a very diverse and hidden ecosystem of particularly insect decomposers at Mygammore. We have no body fossils of insects at Mygammore. We do have fossils of those carnivorous gastropods, as I mentioned, but we don't have any body fossils of insects. Now, we knew kind of going in that insects should probably be around. You know, we find them at other Jurassic sites, but not every fossil site is conducive to preserving every type of fossil. Insects are very delicate. They require very quiet, very gentle conditions to preserve. They're common in places like Douglas Pass, where you have the right conditions in the Green River Formation to preserve their delicate structures. But Mygammore, which, you know, would have been a watering hole during the wet season where you have lots of dinosaurs trampling about, churning up the muck, pushing bones down into the ground that are still hanging out on the surface. That's not a good environment for preserving fossil insects. But now we know that that insects were around at the site, and we know what types of insects, even if we don't know exactly which one it was, were around at the site, and that they were really abundant, that they were very dense. If the insects that get all the way down to the bone in a carcass were as abundant as the ones that we're finding at Mygammore, imagine how thick with maggots the fleshy part of the carcasses would have been. Just gross. If you're ever going back to Mygammore in a time machine, take a hanky to cover your nose because it's going to smell really bad. And you might even catch a glimpse of Allosaurus cannibalizing its own kind. This was the first evidence that we were able to supply the scientific community with, that Allosaurus was prone to cannibalism if stressed out enough. Cannibalism has been shown in other dinosaurs like McHugh and T-Rex, but never before in Allosaurus. But now we know that if you stress out an Allosaurus enough, it will eat its own kind. And then finally, we are finding so many more marks at Mygammore Quarry than we ever expected to find, both invertebrate traces and in theropod bite marks. This is really weird, and we don't really know why it's so weird. There's two possible explanations why our percentage of bite marks is so much higher than any other fossil site that's been looked at. One, Mygammore is just weird, and it's the perfect site for preserving theropod bite marks. That's why they're so abundant there. Or, and this is the one I'm kind of leaning towards, bite marks have been underreported. Insect traces have been underreported from other fossil sites. Typically what you get is, I found this one bone, it had a mark on it, and that's the research that's presented. These site-wide large-scale surveys like we did in this study are pretty rare in Mesozoic deposits. And what we want to do is we want to take our method and apply it to some other dinosaur-dominated fossil sites in the Jurassic period and see if we get similar numbers at Mygammore. And maybe what we're finding is evidence of sampling bias, of people not identifying these marks, or maybe Mygammore really is an unusual fossil site. But we won't know until we perform the same methods at another site and compare results. So that's going to be the next phase of this research. And then finally, and this is probably the most meaningful conclusion, probably the most broad-reaching conclusion that we have been able to draw from the study, it's very helpful to reevaluate your collection protocols from time to time. Now, this seems like an obvious thing, but it's very common for people to have a method that works for them and to just always use that method. But reevaluating your collection protocols from time to time, looking for different types of data, not just looking for the data that you've always looked for, can help you collect more information and give you a better understanding of what was going on at your fossil site than just collecting the beautiful, pretty display quality specimens. Try collecting some of the ugly bones too. The nugget bucket method is pretty cheap. It just requires some Sharpies, maybe a plastic bag or two, and some buckets. It's really easy to do a couple years at a site of collecting those nuggets and seeing if you find any additional marks. And then you'll know whether or not you're capturing all of the data from the site, or if you're only capturing some of the data from the site. But you'll never know which of those two until you take a look. So we're trying to get the word out on the utility of nugget buckets and how they can be effectively used at other fossil sites. And that'll be another part of our research going forward, is getting the word out on that method. And so finally, lots of people to thank. It's been over 30 years of excavation at the Mygammore Quarry by both the Museums of Western Colorado and the Dynamation International Society. And we cannot thank the volunteers, the field crews, and the families that have joined us over the years enough for making this research even possible. My co-authors are amazing people and super resilient for spending so many hours, so many days in a collections room, trolling through mounds and mounds of fossils with flashlights looking for marks. Big shout out to Brandy Mayer, who helped us enter a lot of our research into spreadsheets. And of course, Brian Ng for supplying the wonderful paleo art that you saw in this talk, as well as that animation. And a huge thank you to the Bureau of Land Management, and happy birthday to the McInnis Canyons National Conservation Area. Without you guys, we would not have a talk to present to you today. This research was funded by the David B. Jones Foundation, and big thank you to the Colorado Canyons for inviting me to come share our research with you guys here on this virtual webinar.
right, well, thank you so much, Julia. I know I learned a lot, and I'm sure others did as well. So we'll start taking questions now. We have a couple that have already come in, but if you do have questions, you can type them into the question and answers box on the bottom of your screen, and we'll just start going through those. All right, so let's see. Our first question is from Sally. Why was the quarry such a great spot for dinosaurs?
So the Mygammore Quarry is kind of the best place to become a fossil. You're near a waterway where a lot of animals are living, so you're in the area where they are already. And you have this sort of slow accumulation of mud from seasonal floods that get onto the floodplain and from animals constantly living and dying in the area. So you're sort of in their own natural environment. You do get occasional, carcasses washed in from elsewhere, but you're in the right area for animals to be. You're in the right area to preserve a lot of bone. With a high water table, you get a lot of groundwater coming through the soil, carrying dissolved minerals, which help to fossilize the bones.
All right. Our next question is from Alejandro. Could the giant unknown theropod at the quarry be a Torvasaurus?
Torvasaurus is one of the animals that we are considering. Torvasaurus has a little bit different teeth than Ceratosaurus and Allosaurus. We would have hoped if it was a Torvasaurus, we know we have Torvasaurus at Cleveland Lloyd and at the Dry Mesa Quarry. We would have hoped that this animal, whatever it was, would have shed some teeth along the way, but we don't have any teeth that would have gone with this animal. But we are considering Torvasaurus. We're also considering animal the size of Saurophaganax, but the marks aren't indicative of a species level or even a genus level identification. The best we can do is identify a body size, and Torvasaurus would fit right into that as well.
All right, next question is from Nate. Are there any excavations planned for the remainder of the year?
No, we are not doing any excavations for 2020. We are planning to reopen the quarry in 2021. We had to cease quarry operations due to COVID this year, and now we're getting into the fall, which is when I'm back at CMU teaching, so not a good time to open up the quarry. But we are planning to reopen the quarry in 2021 and resume excavations.
Okay, next question is for Michael. Is there any way to figure out what variety of insect or other invert left those mystery marks that you haven't already tried?
The only other thing that we can possibly do at this point is to find a Jurassic insect quarry. And that's easier said than done. But Jim Kirkland and his colleagues just recently published a paper from a quarry over in Utah where they found some Jurassic insects. So my hope is that other researchers continue their very important work of finding fossils, and we will eventually find some insects that will match up with those weird unknown actors at the Mygammore Quarry.
Okay. This next one came in during your lecture at 6 o'clock. How did you figure out the invertebrate trace marks? Were those the things that compare to the modern beetle markings you talked about?
Right, so once we started finding something that was that small of a size, it's smaller than anything that a dinosaur would have made, and especially those marks that have paired scrapes coming at them, those are very common of insect mandibles, right? They have those pinchers that come in. So we started looking at the literature, we started looking at some of the publications from the forensic literature as far as, CSI work and all that sort of things like body farms and that sort of thing. And that's what really got us into the invertebrate world of, just using that comparative diagnostic anatomy to try to match up which invertebrates were most likely to correlate to which traces.
All right, so the next question we have is kind of a multi-part question from Graham. The first question in it is, were you able to examine any mandibular bones of the theropods?
So we have hundreds and hundreds of teeth from the Mygammore Quarry. We do have some jaw fragments. We did not find any bite marks on the jaw fragments, but you know, we tend to find most of the bite marks on postcranial material just because cranial material is so rare. But we did find bite marks on parts of the skull like the lacrimal and other parts at the top of the skull, but we didn't find any bite marks on the jaws of other theropods.
Okay. We also have a few questions coming in from Ken. Let's see. Is it possible that the evidence of unknown actor number 1 could push back the known presence of insects by 15 to 20 million years?
So we don't think so, and we don't think so because of the way the origin of their groups was determined. It wasn't just determined by fossils, it was also determined by DNA. So what they did, what molecular scientists have done with the wild bees and the burrowing wasps, is they used substitution patterns in their DNA to estimate how long it's been since they diverged from other groups. And the molecular analyses put it into the Cretaceous period, and the fossil evidence also put it into the Cretaceous period, which makes it unlikely that both independent lines of evidence have misjudged the origin of those groups.
All right, and Ken's second question is, will you do more insect research at the Fruta-Paleo area?
You know, we want to expand this research to other fossil sites, and the Fruta-Paleo area is right outside our doors, so we're not opposed to that.
Awesome. All right, let's see. Sandra is asking, who would we contact if interested in joining a dig?
So we will publish our dig schedule before the end of the year, and you can find it on dinodigs.org on the museum's website.
Awesome. All right, any other questions from folks? We have a lot of feedback coming in saying thank you for the amazing lecture.
Thank you for having me. It's always fun to talk about weird and wonderful things that you've discovered, even if they're slightly gross.
All right, it looks like we have a question from Cam. First off, thank you. You mentioned the frequency of feeding traces being unusually high. Could another possible explanation for this be due to prolonged exposure to the surface before being buried, or having several episodes of reburial causing the high frequency of feeding, or possible paleogeographic influence?
So that's something we do want to look at. So we know that we have slow deposition at Mygammore. So our first order of business as we expand this study to other sites is to look at other shale and mudstone deposits that are also slow deposition. So if those two things correlate with their frequencies, then we can say something about, you know, maybe it's the exposure time and access to the remains. But we also want to compare eventually our slow depositional environments to something that's more rapid. We would love to look at a rapid burial scenario like up at Carnegie Quarry at Dinosaur National Monument, relook at that site with our method and see if we see the same thing or not, if we see a decrease in the frequency. But we won't know that until we start looking at other sites. But that is a research question that we're very interested in finding out the answer to.
All right, great. Well, thank you everyone for the questions. That's about all we have time for. And then just to close things off, if you are interested in learning more about CCA, you can visit our website at coloradocanyonsassociation.org for more community events throughout this fall. We do have 2 more lectures, part of the Life Science Lecture Series coming up in October. Our next one is going to be October 6th at 5:30 again with Dr. Aparna Palmer from CMU, and she'll be talking about water bears of the Colorado Canyons. Really cool creatures that a lot of people don't know about living in our canyons. And then the final one will be October 20th, also Tuesday at 5:30. With Dr. Nikki Grant-Hoffman, who is the ecologist of the NCAs at the BLM, and she'll be talking about some recent fire restoration treatments going on in McInnis Canyon's NCA. So thank you all so much for joining. I'll send an email tomorrow with that information and also with the recording of this lecture if you want to watch it again or share it with friends. You'll get that link tomorrow. Once again, thank you, Julia, for being here and talking all about your research. I think everyone found it very interesting. So with that, we'll go ahead and end the meeting, and please feel free to reach out if you have any other questions. Bye, everyone.